trust along with research grant award ranging from 10,000 to 25,000 rupee 1,000 in the past 1 year only. Her topic for today's talk is HPLC in diagnostics and research. HPLC, that is high performance liquid chromatography. It's a powerful analytical technique used in both diagnostic laboratories and biomedical research. It enables the precise separation, identification, and quantification of components in complex biological mixtures. And now I hand over the mic to Sonal Sangwa. Now, please, Sonal, please put the light on HPLC in diagnostics and research. What are its advantages there? Please.
Sure. Sure. Thank you so much, sir, for the nice introduction. A very good evening to my seniors, my colleagues, and dear students who have joined us today. Sir, am I audible, sir? I just want to confirm once.
Yes, you're audible.
Yes. Okay. Thank you, sir. Thank you. I wish to thank ICMLS for providing me this opportunity to present a bit of my knowledge on the topic, as sir aptly explained, as part of week-long NMLPW celebrations which are going on nationwide. So without further ado, I would begin with my topic, which sir already described, is from principles to practice, HPLC in diagnostics and research. So why we think analytical technologies matter in medicine and biology? Of course, they play a vital role and they are very crucial in medicine and biological fields for these particular reasons, as they play an important role in disease diagnosis and monitoring via early detection, personalized medicine. Once the disease is diagnosed, we may tweak the dosage and medicine for a particular patient and monitoring its further treatment, and thus gaining some effectiveness into that particular treatment. Secondly, in the development of drugs, from drug discovery to formulation, their quality control, studying the pharmacological kinetics and pharmacodynamics of the drugs for further improvement in the area. And this all leads to our better understanding in the biological processes, which further ranges from molecular biology to the level of proteomics, study of proteins, study of metabolites via metabolomics, and genetic analysis. So let's put some light on key analytical techniques that we have already known from a long time now. First one is spectroscopy, which analyzes the molecular compositions by measuring how molecules interact with light. So these further have many features like infrared, UV, visible, and Raman spectroscopy, which forms the basis of all these techniques. Secondly, we have chromatography, which is the separation of complex mixtures of molecules. And this further allows for their individual analysis via liquid chromatography, gas chromatography, etc. Mass spectroscopy, which is a later advancement in these fields, as it identifies and quantifies the molecules by measuring their mass to charge ratio. So it further goes into the depth to the smallest of the molecules that we can target further. Next is electrophoresis that we are already aware. It can be of capillary, it can be of gel, and further and so on. Next is microscopy. This allows for the visualization of biological samples at cellular and molecular level via fluorescence microscopy or electron microscopy. So all this leads to why particular HPLC or high performance liquid chromatography. What importance does it holds in today's scenario? Now, it has become very vital analytical technique for separating, identifying, and quantifying the components in particularly liquid samples. It is a versatile technique as it is employed in various fields like pharmaceuticals, environmental analysis, and food science. It excels at analyzing compounds which are not otherwise easily accessed or are very stable otherwise. For all these already mentioned techniques, they cannot be quantified with much specificity and sensitivity. So these all properties have gained HPLC its significance in today's scenario. So this particular talk, the learning objectives I am aiming today is includes understanding the principles of HPLC, exploring the diagnostic and research applications, some of it, and what advancements and future trends are holding for HPLC in the future. So going a bit back to the history, looking back, we have this gentleman, Michael Sweat, who was a botanist. He was the one who first discovered the concept of chromatography uh, by uh, extracting the plant pigments. What he did was he did nothing, but he just crushed the leaves of some plants and mixed them up with petrol or ether ethanol mixtures and just passed through a powder of chalk. This was a chalk powder, or as we call calcium carbonate. And what he received were the multiple colored bands in the form of chlorophylls and carotenoids. So this was the first idea of chromatography that we have known today and developed further into various techniques. So chromatography, now we know that it is a separation technique and it relies on the differences in the polarity, size, and other physiological properties like solubility, charge, hydrophobicity, etc. As they partition between the mobile phase, which is carrying along the sample, on the stationary phase and is causing the separation of the analytes. Now, uh, after the discovery of chromatography, certain principles of chromatography were observed with major breakthrough by these scientists, Martin and Sinch, who received a Nobel Prize in Chemistry also in 1942. They gave us a theory which was called plate theory, that how the components of a mixture are being separated via mobile phase. So this, according to this theory, um if we imagine the column as a ladder and we are climbing the ladder, these lines resemble these stairs. So these were called theoretical plate, and they were of the idea that the analyte in the mixture when passed through mobile phase, they kind of strike an equilibrium between stationary phase and mobile phase, which forms the ultimate illusion at different time scales in the stationary phase, and the illusion of the components happen. So for this, they gave the formula also, which was plate count or number of theoretical plates is equal to length of the column divided by the height of the plate. So within each theoretical plate, analyte is completely equilibrated between stationary phase and mobile phase. So the number of theoretical plates is like number of steps. So a column, by this formula, we can see that a column with more plates or more steps will lead to more efficient separation or an easier climb up the stairs. So HPLC is, again, if we look back and explained already, it is a chromatographic technique which is separating mixture of compounds in analytical chemistry and biochemistry with the purpose of identifying, quantifying, or purifying the individual components of the mixture. But what is the difference here? It is high performance or high pressure. Here we are employing a high pressure pump, which is pumping the mobile phase into the column and causing the separation at much faster speed. So why HPLC? What are the uses and what are the advantages? So it simultaneous analysis is happening, which is leading to early recovery of the separated components. It is very sensitive, as resolution or the separation of the analytes is very good, and the separation capacity thus is very good. So very less time taking and very efficient. And the reproducibility, accuracy, and precision is also very high. We can easily visualize the separated components in the form of peaks. And further, we can also collect the fractions. As the particular analyte from the mixture is passing, we can actually collect it and then purify it, or then further send it to for the further analysis. So it is also very important for the validation of the products in case of pharmaceutical industries. So now let's see how HPLC separates. What are the principles? So we have mainly two types of phase separations. One is normal phase, second is reverse phase. Let's see what are the differences between these two phases, or what are the main features of these two types of chromatography. In normal phase chromatography, the stationary phase is polar. That means it is more hydrophilic in the form of silica or alumina. And the mobile phase is non-polar, like hexanes and chloroforms are used. And in reversed phase, the stationary phase is non-polar, which is hydrophobic. In this case, we use C18, C8, these kind of columns, which are bonded to silica in the form of carbon chain. C18 here denotes that 18 carbons are there. And polar mobile phase is employed in reversed phase. That includes water, methanol, is acetonitrile. So the normal phase chromatography is very helpful in the separation of lipids, saccharides, and certain vitamins. So, and reversed phase chromatography, it is widely used or accepted as it is very versatile, and it separates a wide range of compounds, including pharmaceuticals. So its applicability and reproducibility is high. Another is size exclusion chromatography and ion exchange chromatography. In ion exchange chromatography, we know there are ion exchangers. So positive and negative cations and anions will attract. So here cations will be the ones who will be passed earlier, while anions will be coming later, as there will be attraction between cation and anions. Similarly, in case of cation exchanger, the anions will be the one who will be passed earlier and cations will come later. So mechanism, we can say that it separates compounds based on their ionic charge. Stationary phase here includes the charged functional groups. They can be cationic or anionic depending upon the our molecule of interest that we are looking for separation. Here, mobile phases are aqueous solution with appropriate pH and ionic strength, which is the main requisite for this kind of chromatography. And size exclusion chromatography, it separates compounds based on the molecular size and shape. So here, porous material allows the smaller molecules to penetrate the pores. So the smaller molecules will be penetrated into this porous material, and the bigger molecules or the larger sized molecules will be the ones who will be passed quickly. So in this way, separation happens in size exclusion chromatography. Sorry, I have written wrongly. This should be ion exchange and this should be size exclusion chromatography. I'm very sorry for this error. Now coming on to the schematic of HPLC. So there are majorly four parts that forms the instrumentation of HPLC. First is solvent reservoir. Second is pump, which will pump this solvent into the HPLC column. Third one is our stationary phase or HPLC column. Here, sample will be mixed from the solvent. Sample will be mixed and will be injected into the HPLC column via high pressure pumps. Fourth one is the major part, which is the detector, which will detect any changes in the wavelengths of the molecules, and they will be visible on screen via the software that we use with the particular instrument for the data acquisition. So let's take up all these parts one by one. First is solvent or mobile phase reservoir. They are the glass bottles. Obviously, they have to be inert so that they don't react with a mobile phase. So glass, high quality glass bottles are preferred. Stainless steel were used earlier, but now more or less, we use glass bottles that can hold up to 1 liter of mobile phase. As I said, it should be inert to a variety of aqueous and non-aqueous mobile phases, because ACN or acetonitrile, methanol, water, and buffer mixtures are usually used for the mobile phases in various types of separations. Next is in the part of mobile phase, there is a very important precautions that we use. We always filter the mobile phases via disk filtration, or we use disks, different types of 0.22 microns or 0.4 microns we usually use for the filtration of mobile phases. And then we take out any dissolved gases or air that is dissolved in the process. So we sonicate via high frequency sonicator. So these two steps are vital before we inject the mobile phase into the system. So this has to be compulsorily done at every time whenever we switch on the instrument. Next is high pressure pump, another very important part. This is the high pressure pump where the solvents or mobile phase are passed through the column at very high pressures. So primary requisite is that it should be able to sustain a very high pressure, mostly 1200 bars, which is very high. So as the particle size of stationary phase is smaller, within 5 to 10 microns, the resistance to the flow of the solvent is high. So we have to take care that mobile phases and everything is of very high quality, and they should not carry any impurity, because the flow rates can range from 0.1 to as high as 10 ml per minute. Now the third part is stationary phase, which is the replaceable part. It is not fixed to the instrument. We can change this stationary phase according to our analyte, choice of analyte, according to our chemistry that we are aiming. And different types of stationary phases come in the form of these type of columns. So actual separation is carried out here. Stainless steel or tube size ranges from 25 to 100 centimeter, and internal diameter, this cross section is 4.6 mm, 2 to 4.6 mm. This column is filled with small particles. As I said, it can be silica, and various aliphatic chains can be linked to it on these base. And the separation is the result of different components adhering within this packing in the particles when the mobile phase is forced through the column. This is the cross section of the column that we can see. Peak, this is inert material which actually protects the inner packing. It is, I think, polyether ether ketone or something like that. And stainless steel, outer covering is of stainless steel. Inner, this is peak. And this is the tightly packed material. So for the longer life, we have to take care that our mobile phases are good, and we have filtered all our samples before injecting into the system so that we can maintain the integrity of this inside of the column. So this is again figure, this is the frit, this is the actual column packing, and a certain direction is given for the flow. That means we have to keep a same direction to maintain the life and efficiency of the column. Next important part, fourth, is the detectors, from where actually we will be able to analyze our analyte which passes through the column. So we have wide range of wavelengths ranging from UV, visible, and fluorescence. So they can have different features according to our different requirements, different needs. So they can be able to analyze on the basis of absorbance. They should be should be able to analyze on the basis of refractive index. They should be able to analyze on the basis of fluorescence. It should be able to analyze on the basis of electrochemical differences and light scattering. So taking a look at the UV visible detector, which is the widely used detector, as it detects large amount of compounds because most of the drugs have appropriate structural characters for light absorption. So here, mostly tungsten filament lamp is used, and it allows a range of wavelengths to be detected simultaneously. So we obtain a kind of, if we don't know any wavelength of a particular compound, say we don't know what we are looking at, what all would be there in the sample. So we can give a whole range of spectra ranging from UV visible, that is less than 400 nanometers, till IR, more than 700 nanometers, and visible is around 400 to 700 nanometers. So for that, diffraction gratings are frequently used. So this kind of a 3D spectra we can obtain, and we can look which is the optimum wavelength so that we can pick for a particular analyte. This is the peak intensity, and these are the runtime. Say we have given runtime for 4 minutes. So within 4 minutes, these all peaks are there, and on the basis of area and height, we can look into the intensity of a particular peak at a particular wavelength, and that's how we can select from this wide range of wavelengths. Another type of detector, and it is the most powerful in terms of detection and sensitivity, it is the fluorescence detector or fluorimetric detectors. So here we use xenon lamps. Xenon lamps are used. We know that fluorescence, if a compound is fluorescing, it means that it is emitting light at certain wavelength, when a compound returns to the ground state after transitioning to the excited state. So here we use two filters for two wavelengths, excitation and emission. Once the compound fluoresces and it releases energy, so the second wavelength is used as for the emission of the light, and then we receive the signal. So the excitation source passes through the flow cell to a photodetector, while a monochromator, it measures the emission wavelength. And as I said, it is very sensitive and specific. This is how we receive the chromatograph on our screen, how we visualize on our screen. This kind of chromatogram we receive, or a graph we can say. On y-axis, we receive the arbitrary units in the measurement of signals. And on x-axis, there is time, say for 5 minutes or 10 minutes, we have run a particular injection. This is the dead volume, or the volume that is stored within the column, permanently stored within the column, or the tubings, etc. And the time, the moment when the particular analyte passes from the stationary phase and enters the detector, it is seen in the form of a peak. So this kind of a peak we receive, then we can say that something is happening inside, or we have received some signal. So we receive such kind of a peak. We are able to see this, and this is the height and complete area. And the time at which it is detected by the detector, it is called retention time. And we can quantify our component by comparing it with a standard curve on the basis of the area and height received by the peak. Another important aspect of method validation can be that how well two illusion peaks are differentiated in a chromatographic separation, because it is all about method development, how we have developed our methods so that we receive segregated peaks from the mixture of sample. It can be any matrix. It can be a biological sample like blood, urine, or some fluid, body fluid, or it can be an environmental sample, a food sample. So we have to quantify uh, and see what peak or what analyte we have received with respect to standard. We should be able to separate them properly so that it doesn't get clubbed from base to base. We see here from base to base. So this is a high resolution example of a chromatogram where we can see three separate peaks very well differentiated. And this is a moderate resolution as two peaks are getting clubbed. And another one is low resolution. Here we can see A and B, they are kind of merged, and it will give a shoulder whenever some of the component in any sample is increased. So it might become a single peak. So we will not able to identify it correctly whether it is, and it can give false results also. So method development is a very important aspect, and resolution and sensitivity and reproducibility all play a very important role in HPLC analysis. Now, coming on to the applications of this technique, HPLC is widely used in case of drug discovery. Proteins, peptides, nucleotides are very well differentiated and identified from the samples. Clinical analysis like polystyrenes, phthalates, which are environmental contaminants, they can be identified and quantified. Pharmaceutical application is a major aspect, as drug metabolism through drug metabolism study of antibiotics and certain hormones, drugs, etc. We can see what are the levels in the patients and accordingly adjust the dosages in the patients. Environmental chemistry as to see the environmental contaminants in particular samples like indoor air pollutants, which are polyaromatic hydrocarbons, isopyrenes, naphthols. These are all indoor air pollutants from cooking, etc. They are all released and they are everywhere that we cannot see, and the and unseen harm that they are giving us. Inorganic ions, herbicides, pesticides that have entered now in the food chains. So this all environmental chemistry has now become a very major area of interest, and HPLC is of great help in analyzing such components. Diagnostic studies where we can analyze amino acids, vitamins, homocysteines, In case of this is genetic disease, in some cases homocysteines are very high. So all these applications are there in HPLC. Now, particularly coming on to the diagnostic applications where it is being actually being used. So qualitative and quantitative analysis both is being seen in case of clinical chemistry, where cortisol, sex hormones like peptide hormones like insulin growth hormone, amino acid derived hormones, thyroid hormones, catecholamines like epinephrine, non-epinephrine are being quantified through this technique. Also, fat soluble vitamins like A, D, E, and their metabolites are also being analyzed through HPLC. Newborn screening, analyzing of amino acid and acyl carnitines, drug monitoring, therapeutic drug monitoring, and in case of toxicology studies also, pesticides exposure, etc., are being monitored through HPLC. In case of infection and inflammation, detecting the level of biomarkers and cytokines are also very good help in case of HPLC. And cancer biomarkers, which is a very important field, and it is of very interest in terms of research. When HPLC, when combined with MS or mass spectroscopy, it is being used to discover novel biomarkers in case of gastric cancer, lung, ovarian, breast cancer, etc. So all these diagnostic applications are there of HPLC. Now, what about research? What is being done in research? HPLC is of great help here also in case of proteomics and metabolomics, in case of compound profiling, and when coupled with MS or mass spectrometry, we can further break down the compound and look at a particular ion on the basis of mass to charge ratio and quantify it and identify it, that what, and then the pathways can be elucidated out of it. Another is food and environmental research. Toxin analysis, contaminants can be seen in different kind of samples. Nucleotide DNA adducts can also be seen. DNA adduct, as we know, are formed when a chemical compound, certain chemical or genotoxic, some compound is genotoxic, and it is directly binding to DNAs, and they can be potentially lead to mutations and can contribute to the development of cancer. So yes, in case of cancer risk evaluation, biomarkers can be developed using HPLC and is being in the research application area. And pharmaceutical studies, of course, it is of very high importance in case of pharmacokinetics. Every pharmacological lab has HPLC instruments and they run round the clock without stopping for 24/7. Now let us look at some of the case examples as to where we are using it. Now, environmental contaminants or pollutants that get released into the environment and enter into the food chain, we are all aware of, we all listen to that plastics are being the major culprit nowadays. All the chemicals coming out of plastics like phthalates, bisphenols, not just BPA, other analogs also of bisphenol are found to be of equally harm. As BPA is harmful, another analogs are also very harmful. Dioxins, flame retardants, phthalates, these are found in toys and microplastics, etc. Pesticides, phytoestrogens, or phytoestrogens are, I mean, they are the sources of endocrine disruptors, these all. This is polyfluoroalkyl substances, PFAS. So they are the silent contributor to the metabolic diseases nowadays, because these diseases or these conditions are on rise, like diabetes, hypertension, and such metabolic disorders. NAFLD, non-alcoholic fatty liver disease, every third person is having it, even if we don't know it. So these are all found to be contributing to all these metabolic syndromes. So it is very important to, at a basal level or at a community level, we can able to have a robust method which can actually separate such components out of the body samples or the environmental samples that we receive. So endocrine disruptors are that they mimic our estrogens or testosterone and etc. So they compete with these hormones and they enter our body systems and can disrupt the hormonal balance of our body and can lead to severe diseases. So first such method is HPLC FLT based, which was found in the canned vegetables. BPA was nicely separated in the mixture of canned vegetables, which was found to be very low at a, it could be detected at a very low concentration of 0.005 mg per kg. Another is pesticides, which were found in the drinking water from the farm fields that composed of these particular elements and components. This was the method that was employed, mobile phase of water and ACN, which is not a very, I mean, this is a very common mobile phase that is used. And at a wavelength of 214 nanometer, they were able to separate with as smaller as 1 microliter of the injection. Another is methotrexate. Methotrexate, many of us might be knowing.
Miss Sonal, please stick to the time.
Right, sir, right, sir. Just winding it up. So methotrexate is one of the gold standard line treatment for rheumatoid arthritis and cancers also, but it is not detected in the serum sample after 24 hours, say. But it was found that when it was bound to the polyglutamates, and then it was able to be detected. It was taken up by the RBCs, and they were able to see these particular polyglutamates in the sample. So such kind of drug monitoring has helped the doctors to tweak the dose to minimize the side effects of the drug. So this is our study. I mean, it is the PGI-MER study, so which is now being employed on a regular basis. So coming on to the advances and challenges. So advances include that we have to able to further minimize the time by using ultra performance liquid chromatography, which has reduced the time and solvent use considerably by 1/3. So this is micro and nano HPLC. And of course, when HPLC is coupled with MS, its efficiency is increased. But of course, there are challenges also. It is very costly. It comes at a very high cost, and its complex. Its operation is complex, and high technical skill requirements are there, and excessive sample preparation is also there. If you look at the future trends, it looks really appealing, as the integration of artificial intelligence to optimize and enhance the HPLC analysis is being looked into. The development of miniature portable systems for the on-site analysis, and a growing focus on green chromatography practices to minimize the environmental impact. So we have gained fresh insights, as it is the cornerstone in modern diagnostic and biomedical research. And from biomarker discovery to therapeutic monitoring, HPLC has enabled very accurate and reproducible and sensitive analysis. So these are some of the books that we can refer, or if students are there, if they want to search more and read more about it. And yes, we hope that from bench to bedside, HPLC delivers. Thank you so much for your patient listening.
Thank you, Ms. Sonal,
Thank you, sir.
for nicely explaining the HPLC, the working principles, and the applications. We can take one or two small questions if there are. I request audience.
Sure, sir.
If there is no question, then I think we can stop it because time is going up.
So I can stop sharing now, sir?
Yeah, please, you can stop sharing.
Thank you, sir.
Now I request to hand over the mic to Mr. Chandra Prakash. Mr. Chandra Prakash is a medical laboratory technologist at Department of Blood Transfusion, PGI-MER, Chandigarh. He is BSc MLT from Manipal University, and he did his master's in transfusion medicine at PGI Chandigarh. And before PGI, he served private organizations for I think 20 years, which include Fortis and Vaxi Medical Laboratories, then Manipal Institute of Regenerative Medicine, Sri Gangaram Hospital. His core interest is immunohematology and blood banking, and he has attended and present papers in more than 10 conferences. Now I hand over the mic to Mr. Chandra Prakash to start his talk on just hello to start his talk on transfusion transmitted infections, the practical aspects. Definitely, this is very important topic in daily routine. Many hospitals, many blood bank centers, they take the blood and then they give it the blood, transfuse the blood to the patients, and there are many practical aspects which need the proper looking into so that blood transfusion infections do not occur. So I request Mr. Chandra Prakash to explain the topic. Mr. Chandra Prakash.
Hello, sir. Am I audible? Good evening, sir.
Good evening.
Good evening. Good evening. So, he has asked me to share the slides. Give me a minute.
Okay, sir.
Yeah. Is it visible?
No, not yet.
Now?
No. The main meeting screen is coming, but no sharing.
Sharing is not
No
there.
sharing. No sharing of slides.
Just a minute.
Doctor, you have kept a light behind you. Your face is not that visible.
अभी देखने लायक रहा ही नहीं. बूढ़े हो गए.
I would request to be like professional because we are live actually. Like,
अभी स्लाइड शेयर हुई नहीं.
just a minute. Is it now?
Yeah, it's visible now.
Yeah.
Sir,
Chandra.
shall I start?
Yeah, please proceed. Yeah.
Okay, sir. Good evening, all.
इसको
As you know,
फुल स्लाइड कर दो. पांडे जी फुल स्लाइड करो.
It is full slide.
It's I don't think it's full slide.
On Zoom, it is partially looking actually. Like,
Okay.
it's a problem
Okay,
to Zoom actually.
Okay.
okay, sir.
Yeah.
Chandra
Okay,
Prakash,
good
please.
Yes, sir. Good evening, all. As we know, we all are
Your voice is
celebrating.
not your voice. We cannot hear you properly.
Yeah,
Hello.
come close and speak. Yeah.
Okay, sir. Hello.
Yeah, you're audible.
Fine. Okay, sir. So, as we know, we all are celebrating National Professional Week. So, we are given a very nice opportunity to talk on our laboratory technology related topic. And so, I am very thankful to the presenters, to the organizers, to giving the opportunity to talk on the topic transfusion transmitted infection. It's related to blood transfusion banking. Hello, sir. I am audible, sir.
Yeah, you're audible. Yep.
So, so I was provided the topic.
Can we raise the voice?
Yes, sir. I am
Pandey
on
ji, can we raise the voice?
No, we don't have any control over that because the
Okay.
feed is coming from his side actually.
We can hear you very well, but his voice is very low.
Okay.
He's making the presentation through my
Smart.
cell phone actually. Yeah, please.
Okay.
So, as we know, transfusion transmitted infection, we are on daily basis we are encountering in transfusion transmitted infection. So, just a definition wise, we can say those microbial agents which are very important in blood transfusion, which are transmitted through blood and blood products. So those infections are collectively we call it transfusion transmitted infection. So basically, these infectious agents called mild or asymptomatic infections only. So the blood donors, potential blood donors, would be accepted for the donation because they don't have any symptom. So, so after donation, it will pass to the patient. So maybe the patient is immunocompromised or some obviously their immunity is compromised, so they will get infected. So what kind of organism can be transmitted by blood? So it's a large, many type of infections are there. Hepatitis B virus, hepatitis C, malaria, babesia, HIV, cytomegalovirus, Epstein-Barr virus, HTLV, microfilaria, etc. But in our country, those five tests are mandatorily tested for blood transfusion centers. So those are HIV-1 and HIV-2, it's basically HIV, and then HBsAg for hepatitis B, and HCV, hepatitis C, and syphilis and malaria. So we are commonly using screening methods are whether some labs are doing rapid diagnostic with this assay, some are doing ELISA and semi-illumination and PCR, nuclear acid test, nuclear acid based test. So, coming to the rapid test, these infectious agents can be tested to almost all the modules. Like HIV can be tested through rapid testing, ELISA, SEMI, or NAT. But some infections like syphilis can be tested by non-specific tests like BBRL, RPR, Treponema pallidum, like that.
Mr. Chandar, you are
Yes,
not
sir.
audible properly. Please
Okay.
look into if you can raise the voice. We are receiving messages from the audience.
Okay. Is it okay, sir?
Yeah, now it's okay.
Okay. I just connected to microphone. So coming to the rapid test, different type of rapid tests are done for transient transmitted infection. It says we are doing immunochromatography, immunoconcentration, diagnostic or particle agglutination. So one of the important rapid tests is immunochromatographic based test. So it's principally same as the enzyme linked immunosorbent assay. Only difference is that this immunological reactions is carried out on the chromatographic paper. And in the case of ELISA, we are doing on the washers, and we are reading it through the calorimetry. So this is the for the interpretation of rapid test. We all are already familiar with the rapid test, so I am just referring to the rapid test part. And these are the different kind of rapid testing, immunoconcentration and dot plot test. Here we can see in the testing, like in this, we can see the control band, we can see the HIV-1, HIV-2. So for the interpretation point of view, we can see here if the control is comes positive, and then particular HIV-1, HIV-2 comes positive, then only we can validate the test result. So same in the terms of immunoconcentration or dot plot assay. So coming to the generations of rapid testing assays, so it was first generation of testing includes just the antigens derived from the detergent disruption of viruses grown in human lymphocytes. Or so phase-wise, it comes to the recent generation. We used to do the recombinant and chemically synthesized peptides of the virus or virus particles. And now in the recent, we are saying both antibody and antigen. So coming to the advantages of rapid test, we can say it has though some kind of advantages also. As the advantage of speed and sensitivity, results can be read visually. No specific instrument is required, and no calculation is required. Skilled manpower to that level is not required. Highly skilled manpower is not required, and as well sophisticated instruments are not required. But the detection level is poor, or you can say detection level is low. And coming to the demerits of rapid test, so if we calculate the cost per particular kit, it comes costly, because when we are doing the mass testing through the ELISA, it comes very cheaply. And lack of permanent records, we cannot keep records of the card testing, rapid testing. It is not suitable for and the large number of samples. As we know, most of the blood bank having less donations, but the large blood bank we cannot do for the large number of things. And person to person subjective reporting error is also there. So basically, our drug Director General of Health Services, as we know, blood it comes under the drugs category. It is,
मैं किसी मीटिंग चेक कर दूंगा.
so DGHS or National Blood Transfusion Council has formulated a technical manual. It's for serological testing. They say rapid test can be done at a remote blood bank or smaller blood bank. So rapid test is included in the technical manual also. And for the large blood bank, we can use the ELISA and semi-negative based testing method. And for quickly, you know, not very specific test method available. So we are going for non-specific test. Many tests are there. VDRL, we all know, Vendor Disease Research Laboratory test, which is most commonly used test, simple, rapid, and economical. But it has some limitations. The sensitivity is high, but specificity is poor. For some kind of a specific test, if it comes positive in the non-specific test, then for the confirmation, we go for the specific test. So these are lengthy procedures not suitable for routine testing, and the specificity high is not distinguished fast from the current infection. And for the malaria testing, basically most of the labs are using peripheral blood smear, and some blood banks are using quantitative buffet code methods, so it can be semi-automated for this method. And some rapid test kits are also available, and ELISA testing is also available for malaria. For peripheral blood smear, infrared state thick and thin blood cells are analyzed. This gold standard basis, ability to detect quantification also. Sensitivity is up to 500.
Chandra
Prakash ji,
Prakash?
slides are not moving.
Chandra Prakash?
Yes, sir.
You face the, I mean, you have adopted a headset, no?
Yes, sir.
So even now, like your voice is a bit husky. Maybe like that is creating problems for inaudibility. So try to like be, I mean, little louder.
Okay.
Slides are all not also not moving.
He's talking about malaria. Don't worry. Like once he's done with this, I'll move.
Okay.
Okay, sir. So I have to say next test every time. Okay.
Okay.
For next, okay, sir. Next slide. Okay, so jump to the side to malaria, sir. Maybe 6th or 7th slide.
6th or 7th slide.
Okay, okay, yes, sir.
Now this is okay.
Actually, yes, sir. I am scrolling
No.
in my.
It is stick only was the first slide. We are looking only first slide.
Just a minute.
No
Now?
other slide. Now, yeah, it has changed.
Okay.
Okay, sir.
It is on preparation of in-house external controls. Is it okay, or shall I go back?
Go back, sir.
ELISA or malaria?
Malaria, sir.
Okay.
Yes, sir. It's on 16th slide. Yes, sir, malaria.
Yeah. So move on to 16th then.
Yes, sir. Next slide.
Okay. Right?
Yes, sir. Yes, sir.
Continue.
Yes, yes. Okay. So after rapid diagnostics, so the large blood banks are using ELISA-based testing for transfusion transmission. So it is widely accepted. ELISA is a solid phase-based assay technique designed for detecting and quantifying soluble substances such as protein, antibodies, and hormones. In the ELISA, the antigen target molecule is symbolized on the solid surface, on the microplate, and then complex with an antibody that is linked to the enzyme. So this solid phase assay, we are seeing here in this microplate. The microplates are combined with immobilized with the antibody. Next,
Next.
next slide. Yes, sir.
Okay.
No.
Why this is happening, I don't know.
Okay, sir.
Now? Now okay?
Yeah, slide has changed to Chandraprakash. You
Okay.
see to it which one you
Yes,
need.
yes, sir. This slide shows the stages of ELISA. So in initial is adoption of antigen antibodies to the plastic microplate. Then next step, addition of analyte in test samples. Then we incubate, and then separation of bound and free reactants by washing. We just wash out the unbound antigen antibodies, and addition of enzyme conjugate, and then addition of substrate for the color development, and then the reading of the color by colorimeter or system. Then next slide, sir. The essential components are for ELISA, use test kit before its expiry date. It's common for all testing. Each test run must include one full set of controls that yields results within the limits of standard or acceptability. Usually, the kit manufacturer is providing controls, but here in blood bank, we also use in-house control along with the kit supplier controls. So any test run not having at least minimum number of controls within the acceptable range is invalid and must be repeated. Control used the test. Each test run requires a set control to validate the result. These controls must be treated in the same manner as unknown sample. The assay is valid and the results are reliable when the control produce results in the acceptable way. The two types of control we are using in blood bank. One internal, the kit manufacturer provided, and the external, in-house made, usually borderline sample or suppose high range sample, we dilute it to come into the borderline. So that next slide, sir. So here we are just discussing on preparation of in-house control. One control is already manufacturer provided, and the in-house control is a very important part in the blood bank transfusion transfusibility system testing here. So usually we select a high titer seropositive sample, we test, and then heat inactivated the sample, and if plasma is taken, then recalcify it as a serum just you can preserve it for longer time. So then make serial doubling dilution the sample with a seronegative serum. Then we calculate the ELISA ratio. Next slide, sir.
Yeah.
So in the ELISA, main analytical errors are, as you know, in other testing errors are the main analytical errors. Other than this, improper incubation time or temperature, improper washing to remove out the unbound the antigen antibody particles, and the carry over from the adjacent specimen. Suppose one specimen is positive and you just carry over this sample to the next sample. and then equipment malfunction and calculation error. These are the main analytical errors we are encountering with the ELISA testing. Advantages of ELISA is it is suitable for large case samples, as any blood bank are getting donations for more than 100 or 200 per day. So it is suitable for large case samples. Reagents in small volume are required. Colors in product can be read visually. Quality assurance and quality control is easier. Identify serial converter and sensitive when compared to the rapid test. So as we know, the rapid testing detection level is 0.5 to 1 nanogram per ml, and it is less costly compared to the rapid test. Next step.
Yeah.
And after ELISA, one similar to ELISA, we are using chemiluminescence based assay, which is fully automatic. It is the principle is similar to the ELISA, except the chromogenic substrate is replaced by chemiluminescence compound. So it's emits light stretchable luminescence. And like similar to ELISA, we have indirect clear, direct clear. So let's say indirect clear, the testing molecules are not directly involved in chemiluminescence reaction. The excess energy of the molecule transfer to a fluorophore, that is in turn is excited. So basically, CLIA, we are coming to the advantages of CLIA over ELISA. You can say it is fully automated, high throughout, requires less quantity of sample, and the most important is less turnaround time in compared to ELISA. ELISA is widely using, so we are comparing here with the ELISA. It can operate in random assays. Like in ELISA, we used to use 96 watts. Or here we can use for one individual sample also. And the merit in compared to ELISA is high for specialized equipment and few false positives, so the sensitivity is higher. Next step. So as we say, Director General of Health Services or National Blood Transfusion Council guidelines saying for the blood screening of transfusion transmitted infections. If any sample of donor comes non-reactive, then it is okay, release blood unit and fit for use. If it comes initially reactive, then first we have to discard all the blood components of that particular unit, but we have to repeat the test on the sample from the bag and pilot tube if it found reactive for borderline. And then we have to notify to the donor for the second sample. And if second sample is also positive, then we do the donation counseling and refer to the particular clinic. And here the references of the my slide. National Blood Transfusion Committee and Director General Health Services Transfer Medicine Manual and Drug and Cosmetics Act and National AIDS Control Strategy Standard and Blood Bank Policy Manual. Thank you, sir.
Thank you, Chandra Prakash. You have nicely covered the topic. There was a little bit voice problem, but the topic was very nice.
Okay.
Anyway, if there is any question, audience, please. Audience, please. No questions? Yes, please. Hello. Anyway, if there is no, is there any question? Loud voices are coming. Anyway, Chandra Prakash ji, I think we should close the this slide system. Anyway, it was very nice of ICMLS that we are conducting such programs, educational programs, which are just teaching and learning to the students and to the teachers even. And I just suggest that such topics should always be on ICMLS table.
Thank you,
Thank you
sir.
very much. Thank you very much.
Thank you. Thank you so much.
Chandraprakash ji.
Hello. Yes.
Yeah, I think the switch off this slide PPT.
Sharing. Okay.
So thank you very much, and I send you back your mic. It was a nice program, and in future, please keep it up, keep it up. And if there is some topic to be given to me, even during routine, we can do it. So I will be ready for that.
Thank you. Thank you so much. I would, since there are no questions, I'll just wind up. We have been almost there for almost like 1 hour plus. So it was a nice presentation. The first one by Ms. Sonal was really intriguing. Like it took us to the advantages of HPLC all across R&D and diagnostics and uh, production, that is pharmaceuticals. So coming on to Chandra Prakash, my namesake. I am also Chandra Prakash. He's also Chandra Prakash. The only glitch was his voice is basically a little husky. So that is why he was. There were a lot of messages from people who were not able to gather what he's speaking. So next time on, I would just simply tell my friend Chandra Prakash to work on his pitch so that he can be more audible. So take it as a constructive suggestion. With that note, I would wind up the talk. So once again, we'll meet tomorrow with
Chandraprakash,
Hanji.
Chandraprakash, probably he was using some sound system which was not okay. If he worked directly from his laptop or directly from mobile, there will be very good. Your voice was very high pitch. Sonal's voice was very high. I don't know
Actually,
why, but his voice was just like he is speaking from
no,
the
no.
well.
So actually, probably Chandra Prakash would respond to this. I have asked him to unmute.
Yeah,
It was because his voice is husky. Actually, like he was
yeah.
talking. He used both the audio, what do you call, mic systems, the phone as well
Okay.
as the headset.
Mhm.
Chandra Prakash, you have to say something.
No, sir, I just use the headphone, na, sir. That's why I think the voice is loud
Yeah.
and clear.
Okay. Okay. Overall, it was a wonderful
Overall, it
presentation.
was
Yeah.
nice.
Yeah. You're right. Okay then. Bye bye. See you
Okay.
all tomorrow again at 6 o'clock with with some more interesting, intriguing talks and enlightening talks by